Good day, everyone. How are you guys doing? Now, I just want to show you some traits to understand your amino acids, their structures, and their different categories. Like how you're gonna remember everything without forgetting. Okay, okay. Now, in order for you to understand the structure of your amino acids and the different types of amino acids, basically we have 20 main amino acids. That is the common amino acid that does what? 20 amino acids. And these 20 amino acids can be classified into different categories. Are you with me? You can classify your amino acids based on are they essential or they are not essential. When we talk about essential and non-essential, essential means that when you say something is essential to you, it means that that thing you yourself don't have it. You need to get it from what? Something else. Are you with me? Why when we talk about non-essential, it means that what the thing is you can actually provide that thing or produce that thing by yourself. Do you understand it? We can also classify our amino acid based on are they glucogenic or they are ketogenic, or are they both glucogenic and ketogenic. You can also classify your amino acid based on are they polar or non-polar, are they acidic, are they basic, different categories. But with this video, you are going to understand the whole structures of your amino acids. Easy peasy, trust me. So let me just start from the essential amino acids. How do you remember all your essential amino acids? You remember your essential amino acids with this keyword, which is what? Private, team, all. Are you with me? Private, team, all. So you shorten your private to PVT, as in private, team, all. Now these are the what? Essential amino acids. Are you with me? So PVT or P private team all. Now let's count 1 2 3 4 5 6 7 8 9 10. That is to tell you that we have 10 essential amino acids. Out of the 20 amino acids, 10 are essential, while the remaining 10 are non-essential. But mind you, this 10 here is what is for what? Children. Children has what? 10 essential what? Amino acids. In children we have what? 10 essential amino acids. But in adults we have 9. It means that what? You need to eliminate the A, which stands for adult. That's to tell you that what? In adults we don't have this A for adults, right? So in adults we don't have this. This one is for the what? The phenylalanine. This V is for what? Valine. The MT is for tryptophan. So I'm going, like, I'll come back to all this, okay? Please, you guys, I need something to clean the brush. Now that is just that about the essential amino acid, okay? I'm still going to come back to the essential amino acid. I just want to show you that what? Okay, your essential amino acid can just be represented with what? Private team all. That is PVT team all. Now let's go to another category of the amino acids based on are they polar or non-polar. Now let me start with the non-polar, and from there I will start with their what? Structure. How you understand their structure perfectly, trust me. Now we have the non-essential amino acid, non-polar rather. Non-polar, that is they do not dissolve in water. Now non-polar, what are the non-polar? The non-polar are what? How do you remember them? This is the keyword for you to remember them. We have glad, all vampires left the island. So glad all vampires left the island. That is, we are glad that all the old vampires left the island. So the question is, why do you think this vampire left the island? Why did they leave? Why did they leave? They left because they met a professional public health trainers. That is, they need someone on their what? On their health. So they left because they met what? A professional, because they met a professional public health trainer. So the public health can be shortened to what? PH, that is public health trainers, somebody that can help them on their health. You know, as vampires, they need a good health trainers. Okay, so glad all vampires left the island. Okay, why did they leave? Because they met a professional public health trainer. So this glad is for what? Glycine. What did I call it? Glycine. I'm gonna draw the structures. Okay, the Al is for what? Alanine. The third one, which is vampire, that is what? The valine. The Le is for what? Leucine. The next one is Is, which is what? Isoleucine. The next one is methionine. The next one is Pro, which is proline. The next one is PH, which is phenylalanine. Very simple structures. Okay, just calm down. The next one is trainer, which is what? Tryptophan. Okay, tryptophan. Do we understand it? You can please make sure you write it down. Glad all vampires left the island. Why did they? Because they met a professional public health trainer. Now let's start with their structures. Let me start with the structure of glycine. How do we draw the structure of glycine? So glycine is the first guy here. The first guy is glycine. Now let me remind you this. When we talk about amino acids, what are amino acids? Amino acids are basically a type of what? Acid that has an amino group. Do you understand it? A type of acid that has an amino group. So, and basically in organic chemistry, when we talk about acid, we are basically talking about COOH. That is this carboxylic acid. Okay, so amino acid is what? Any acid that has what? An amino group. So we can have a particular compound like this. Let's assume this is a compound, so it has this acidic group. This is the acidic group. This is what makes this compound acidic, right? So what type of acid? An acid that has what? An amino group. So if this substance also has what? An amino group here, this will make it what? An amino acid. That is to tell you that what? This is the backbone of all amino acids. Are you with me? An acid that has an amino group. Don't forget, our carbon is supposed to have how many bonds around it? 4 bonds, right? The carbon is supposed to have how many bonds around it? 4 bonds. So this is what? 1 bond, the second bond. So we have a third bond, which is what? Hydrogen here. And we have another bond here, which is an R group, which is known as what? Our alkyl group. This is no show. No, fine, thank you. Let's continue. So we have our R group, which is an alkyl group. Do you understand this? So that is to tell you that what? The only thing that is changing, you know, when we talk about alkyl group, that is group. When we talk about alkyl group, we are basically talking about methyl, that is CH3. Look here, CH3, this guy, or this, which is what? Methyl. We also have C2H5, that is CH3, CH2, which is known as what? Ethyl. We also have CH3, CH2, CH2, and I'm sure you guys are familiar with this, which is known as what? Propyl. That is to tell you that what? The only thing that will be changing in the structure of our amino acid that we want to draw is just the what? The R group. Are you with me? So the only guy that will be changing is just this amino I mean, this is the only guy that will be changing is just this alkyl group, and that is to tell you that what this one here, NH2, C, COOH, and this H is our backbone for the what? Amino acid. That is why you will see some textbook, they will write it as what? N, which is for the amino group, C, this, and the C, which is for the acid. Do you understand it? Now let's go back to the structure of glycine. Very simple structure, very simple. I said what the structures are, what? Very simple. Now glycine, how do I draw the structure of glycine? Don't forget, glycine is the first guy, and because we want to draw an amino acid, so we need to draw the structure first, right? The structure first, which is NH2, I mean the backbone rather first, C, COOH, right? We have an H here. Now this glycine is a guy, he is the first guy here. Glycine, can you draw an R group here? Can you give us an example of an R group? But because this guy is the first guy, he doesn't know it. He have not even heard of an R group before. He doesn't know what to write. Can you write an R group? No. Okay, just try your best, write something here. Just try your best and write something there. So because this guy does not want to leave the space empty, he will then what? Write this H back. Are you with me? He does not want to leave it empty, so he will just write the H back. I don't know what to write, but just for this place not to be empty, let me write the H back. So this is the structure of your what? Of your glycine. Do you understand it? This is the structure of your what? Of your glycine. Now the next one is alanine. After GLAD, the next one is alanine. The next one is alanine. Let me draw it beside this alanine. The first thing is what? Our backbone, which is COOH, NH2. We have H here. Now alanine is the second guy. Alanine, can you draw this? Can you draw any alkyl group for us? Yes, he knows what to draw. So alanine will then write what? CH3, which is the first group of the what? Which is the first guy in the what? In the alkyl group. So methyl. Are you with me? Methyl. So alanine is fine. It's good to go. Do you understand it? The first guy does not know what to write, but the second guy knows what to write. And what the second guy wrote was the what? The first group of the what? Alkyl group. Now the next one is valine. Now how do we draw the structure of valine? It is not hard. You just draw. You notice that what in this valine, it's just that we have a V and alanine. Valine is just like you having V and what? Alanine. It means that what? You're gonna draw the structure of what? Of alanine and a V. But you just make sure that what? Your V is what? Facing down. You just make sure that what? Your V is facing downward. Okay? Now, so valine is what? V and alanine. Are we together? So our alanine is what? NH2 C COOH. We have an H here, so our alanine is supposed to have CH3 here. Are we together? And we're gonna draw a what? A V. That is what? Facing down. Which will be what? A V like this. This is a V. Are we together? And we have CH3 here. CH3. Do we understand this thing? Now let's look at it. Valine means what? V and alanine. So this one here that I circled in blue color is our what? Alanine, right? And the V, but just turn the V upside down. Are we together? Now the next thing is, let's check this carbon. A carbon should have how many bonds around it? 4 bonds. But the carbon of valine is not this carbon here. I mean this carbon is having how many bonds? 1, 2, 3, and 3 hydrogen, means making 6 bonds around the carbon. And the carbon is supposed to have how many bonds? Just 4 bonds. That is to tell you that what? We need to remove 2 hydrogen from this 3 guy here. Do you understand? Just for the structure to be balanced. It means that what? The normal structure of valine will not be what? NH2 COOH, hydrogen. This CH3 return to what? Ordinary CH. We're gonna remove 2 hydrogens. Okay, so we have CH. Okay, we have CH3 connected to it, and we also have another CH3 connected to it. So this is the structure of valine. Are you with me? So I'm gonna clean all this now. I want to move to the structure of leucine. Don't forget, I said all these things are very simple, easy peasy. Now, one thing I need to tell you here is that what all amino acid, the structure of all amino acid, each of them will have the structure of alanine first. Each of them will have the structure of what? Alanine, and they can then add any other thing to it. Are you with me? Each amino acid will have the structure of alanine, and they can actually what? Add some other things to it. You notice that what? Even this valine itself, it has the structure of what? Alanine, and it's added to CH3 to it. Now the next guy is leucine. Okay, so let me just clean this. I hope you guys have written. Is glad all vampires left island. Why do you think they left? Because they met a professional public health trainer. So let me just clean, so I will have more space to draw the structures. So the next guy is leucine. Leucine. How do we draw the structure of leucine? Leucine is just like valine. Don't forget that what valine is gotten by what? Alanine and what? V upside down. Now leucine is just what? A valine also, but a CH before the word valine. That is just it. Leucine does not know what to write, but you just feel like, okay, I've seen what valine do. Let me just copy valine also, but just add a particular carbon and hydrogen to it, so that I'll be a little bit different from the what? From the valine. Okay, so we have, for the leucine, we have our backbone, or draw your alanine first. You know, that is, you want to draw valine first. Now COOH, hydrogen, NH2. You can write your H2 here. Okay, now just like your valine, okay, but you just add another CH, just like your valine, but you just add extra CH to it. So CH2, CH, CH3, CH3. You know, this is just like your valine. Maybe now the only thing I added is what? CH2 for the structure of what? Leucine. Now the next one is isoleucine. Now, just a trick. Are you with me? Just a trick. Do you know why we have isoleucine? Because isoleucine is just like an isomer for leucine. This guy is very angry. Do you know why? Because this guy was like leucine. You notice that this valine what drew the structure of what? Alanine, and he added its own name to it, which is V. Why can't you just, why do you have to copy the valine? Why can't you just write your own L2? Do you have to copy it? So that is why we have what? Isoleucine and isomer. Do you understand? Now you know why I said it is an isomer now. So this is the structure of leucine. This is the structure of leucine. So isoleucine is actually angry. Why is the guy angry? Because he noticed that leucine copied the structure of what? Valine. And the only thing that leucine added was what? CH2. So isoleucine will be like, no, I don't want that. I don't want that. You should have just done something else. Now let's go to the structure of isoleucine here. Isoleucine, all we have is that what? Our backbone first. NH2, CCOOH, hydrogen. Don't forget, every amino acid, the first thing you need to do is that what? The alanine structure. All of them has the structure of what? Alanine. So write your CH3 here. Okay, now isoleucine. Don't forget the name, isoleucine. What isoleucine will do is that what? It will add what? L to this structure, L, which is from that leucine, and iso, from that iso, it is going to add another what? Another I. That is tell you that what? Isoleucine will add what? L and I. Are you with me? Isoleucine will add what? L and I. So where is the L? The L will just be here. Yeah, let me use the black marker. This is the L. Do you understand it? Let's L. Okay, and I here. Do you understand it? Letter L and I. That is what? Your isoleucine. What do you think we're gonna have here? We're gonna have CH3 here. Okay. Also here there is a particular carbon will be in this middle. You know, there is a junction here. Okay, so a particular carbon is gonna be here. So we have CH3 here too. But now let's look at this carbon, this alanine carbon. You notice that a carbon should have how many bonds around it? Just 4 bonds. But this carbon is having how many bonds? 1, 2, and 3 hydrogen, making 5 bonds. It means that you need to remove 1 hydrogen from here. Do you understand it? Very simple. So let me draw the structure again. To now be NH2C HCOOH. This will now be CH2. That is, we need to remove an hydrogen here. So CH2. This place there will be a particular bond here. We are trying to write L, which will be CH2. Okay, and another CH3, and we have a CH3 here. Do you understand it? This carbon is having how many bonds? 4 bonds. 1, 2, and 2 hydrogen, making 4. So this is the structure of isoleucine. You understand why we said isoleucine is actually angry on leucine? Because leucine actually copied. So isoleucine has to correct everything back. So this is the structure of your isoleucine. Okay. The 3 letter this thing for leucine is LEU. For isoleucine, from iso, I will stand as the isomer, leucine LE. Do you understand it? Now the next structure is glad all vampires left the island. Why did they leave? Because they met. So the next one is methionine. Okay, methionine. So how do we draw the structure of methionine? So we are just going to draw our backbone, then an M. That's all. Your backbone and M from the methionine. So methionine is our backbone. NH2CCOOH. We have an hydrogen here. Are you with me? So the next all you have to do is just your M. Capital letter M. Are you with me? Your capital letter M. So carbon. And what you will do is that put a sulfur here. Okay, carbon. Now let me draw it back. So we have NH2CHCOOH. This point carbon, CH2 carbon, right? CH2 this side. So there will be a bond here. What will be here? S. Bring it down. CH3. And I hope you understand how we got our CH3 here, because this carbon is supposed to have 4 bonds around it. This is 1 and 3 hydrogen. Do you understand it? This is not a double bond here, okay? Just a single bond. This is the structure of methionine. So the 3 code for methionine is just met, which is MET. Met. Okay. Now the next structure is what? What is the next structure? Glad all vampires left island. They met a professional. The next one is proline. But before going to proline, let's go to the next one. Met a professional public health. Now the public health, which is our pH, is known as phenylalanine. So how do we draw the structure of phenylalanine? Even from the name, you have a clue already from the name. That's to tell you that we have a phenyl and alanine. You already know the structure of your alanine before, right? We have phenyl and alanine. And what is the phenyl? Okay, before asking you the question for what is the phenyl, what is phenol itself? Phenol is your what is it called? Your benzene ring, right? And OH. This is phenol. So if this guy is not there, or if any other thing is not there, we have we have our phenyl ring. So it means that you need to add your phenyl to what? Alanine. That means we have alanine. And the structure of alanine is what? NH2CCOOH, right? Hydrogen, phenol, right? But don't forget, you've not draw the structure of your what? Your alanine. Your alanine, we have CH3. This is the structure of your alanine. Then add your phenol to it. Your phenyl rather. Okay. Let me use another marker for the phenol. Okay. So just for you to see that it is what actually what phenyl and alanine. Do we understand it? But mind you, this carbon is having how many bonds now? 5. 1, 2, and 3 hydrogen, making 5. So it means we need to remove what? 1 hydrogen from your what? Alanine. For the carbon to be what? Complete. Means we're gonna have what here? CH2. This is the structure of your what? Your phenylalanine. Are you with me? And the 3 code for the phenylalanine is what? PHE. This was just studying PM, physical and health education. PHE. And the 1 code is what? F, because it sounds as what? F. Okay, it's PH stands for what? F. It sounds as F. Now the next one is professional health trainers. So now the next one is the proline. Actually, the structure of this proline. Okay, so before drawing proline, can I draw some other thing? You understand what phenylalanine is? Let me draw the other guy, because they met a professional public health trainer. That trainer is the what? CR. CR is what? CR. That is tryptophan. Tryptophan. Now this guy is actually telling us something. Tryptophan is telling us something. This trip here is telling us that what? We have 3 things in the structure. And one of the 3 things is your what? Is our what? Our alanine. 1. It means that what? Out of the 3 things, we have our what? We have 3 things here. 3 is telling you that what? You have 3 structures. The first one is your what? Your alanine. Okay. Now what is the next one? Fan. This pH here is telling you that what? We have a phenyl. Do you understand? It's telling that okay, we also have a phenyl. Now what's the last one? The last one is, now let's take it like this. Instead of calling it tryptophan, let's call it a tryptophan. A tryptophan instead of calling it tryptophan. So we say a trip to your what? To your farm. And don't forget that what? In your farm you have a hut which has a single window. So we have a hut in your farm. So we have a hut, right? Like this. That is just like house, but it will have what? 1 window. Okay, that's to tell you that what? We have this structure too. Apart from alanine, phenyl, we also have this structure which is your what? Farm. In your farm you will have a house like this in your farm, but it usually has what? 1 window, and that 1 window is what? Your N, which is nitrogen. Okay, so it means that what? We also have this structure here. Let me draw it. So do you understand? Tryptophan tells us that we have 3 things: alanine, phenyl, and this structure. So how do we draw the structure of tryptophan? Tryptophan. So the 3 short code for tryptophan is what? TRP. Okay. Now let's look at the structure of tryptophan. The first thing is what? Alanine. We have our alanine. NH2CCOOH, HCH3. The next one is your what? Your phenyl. Your house too. Okay, so let's draw the phenyl here. Let's draw the phenyl. You can draw it somewhere here. That is, phenyl is what? Just a carbon ring, right? But it is attached to something. So we have a phenyl, and this your house. You draw it. You can just draw. You can actually draw anyhow, but you can just draw your house facing this side, so that it's gonna take one of the structures from this phenyl. Okay. You can basically draw anyhow, just for it to be easy for me. That is, I took one structure from here so that like they are actually what joined together. So but don't forget that we have a nitrogen somewhere, either here or here. You can actually put it anywhere. So we have a nitrogen. Okay, so this is the structure of your phenylalanine. So you just have to check that. Okay, what do we have to check here? We have how many bonds do we have around this carbon? 1, 2, 3, 4, 5. So it means that what we need to remove an hydrogen from here. So this should be what CH2, so that we're gonna have 4 bonds around our carbon, right? So 1, 2, 3, 4. Do you understand it? Do you understand it? So the next structure is the proline. Our next structure is proline. Our next structure is proline. How do we draw the proline? Actually, if you should just Google search it, you will notice that the structure of your proline actually looks like a P. It looks like an inverted P. That is just the structure of your proline. It looks like this. Your proline looks like this actually. And I can actually turn this thing. I can turn it to be like this, which is still letter P. So it means that when I add the word proline, what I actually want to draw is just a letter P. But before drawing the letter P, I know that I need to have my backbone, which is the amino acid backbone, NCC. NH2 COOH hydrogen. Now the question is, if I want to write a P, do you think I will write my P like this for it to come like this? No, this is not a P anymore. It means that for me to write a P, I need to start from where? The middle. I need to start from the middle and bring it this way. So basically like this, we've actually written a letter P. Do you understand? It means that I'm going to draw something from here to form the letter P. Are you with me? Please make sure you are with me. Stay with me. Now, okay, let me draw it back. Sorry. So we have the NH2 CCOOH hydrogen. So just draw your house. That's all. That is carbon, carbon, carbon. This carbon is gonna have how many hydrogen? H2. You understand now? What about this H2? Do you understand why this carbon is having H2? Because the normal carbon should have how many bonds around it? Just 4 bonds. So 1, 2, and 2 from hydrogen, making 4. What about this? 1, 2, 3, 4. Right? What about this? 1, 2, 3, 4. We are good to go. And that is all about the non-polar amino acids. What about the polar amino acids? For the polar amino acids, we said they are polar. Just have it in your mind that most of them have OH. But how do you remember their name? It is actually one thing I want to, before I move on, I need to tell you this about the polar amino acids. We actually have different types of polar amino acids. When we talk about polar, it is not a must you have charge before you can be a polar. Let me just take glucose for instance. Glucose is actually it can actually be dissolved in water, but it does not have any charge. Okay, so and same as other substances, you don't have to have a charge before you can be polar. Okay, so we have some polar amino acids that are they are charged. We have some polar amino acids that do not have a charge. We have polar amino acids that are polar amino acids that they are acidic in nature. We have polar amino acids that they are basic in nature. We have polar amino acids that they are amide. Are you with me? Are you with me? Now let me show you. Now for the first set that they are polar but they do not have a charge, do you know how we remember their name? You just say set has city, set as city GLE. You know what a GLE is, guys? GLE is called a confirmed Benz. So GLE. So set as CTGLE. But now this is how you are going to do it. You just have to like split the letter into two. Set as CTGLE. Okay. So SE TH AS CT TY GLE. Are you with me? So set as CTGLE. It means that what? For the polar amino acids that do not have a charge, we have SE, we have TH, we have AS, we have CY, we have TY, we have GLE. Is I want to buy GLE. So now look at this. The SE is for the what? For the serine. SE, serine. TH is what? Threonine. AS is what? Asparagine. CY is what? Cysteine. TY is what? That is TY. Which of the amino acids starts with TY? Tyrosine. Okay. Tyrosine. TY. Tyrosine. GL is for what? Glutamine. Okay. Now you understand now. Don't forget I said we have polar amino acids. That of the polar amino acids, some of them do not have a charge. So this one is for those that do not have a charge. All of them are still polar. Okay. They do not have a charge. What about some that have charge? Now one of those that have charge is what? The acidic ones. Okay. We have the acidic amino acids. They have a charge. So the acidic amino acids are, one of the acidic amino acids is, okay, now the code for the acidic amino acid is that what? When we say something is acidic, do you know that what? We boys, we are actually not acidic. To be sincere, guys are not what? Acidic. It means that what? Guys, for the acidic ones, acidic, you say guys aren't, that is, they are not acidic. Guys aren't acidic, which is glutamic acid. After glutamic, we also have glutamic acid. We have aspartic acid. So glutamic and aspartic acid. Do you understand this? Glutamic, aspartic. The next one is what? The basic one. The basic one are how do you remember the basic one? You just say, okay, for the basic ones, for the basic one, you say is lies. Is lies. You can see the way I spell my own lies. Is lies is basic. Is lies is basic. Okay. Is for what? Sorry, is lies are basic rather. Is lies are basic. So this one is for the histidine, lysine, and the arginine. Okay, so these three guys are what? Basic. Why these two guys are what? Acidic. But these two guys are also what? Polar. It means that what? These are the guys that are what? Polar and still have a what? A charge. Now, before I move to the structure of this asparagine and GLE, let me just draw the structure of this CY and TY. Don't forget that what your CY is your what? Cysteine. CY is for what? Cysteine. So how do you draw the structure? Just alanine and S from here. Alanine and add this S. That's just your structure. NH2. Let me use the other marker. Sorry. NH2 COOH hydrogen H and no. The structure of your alanine first, sorry. The structure of your alanine, CH3 and SH. This S is coming from where? From this system. Do you understand it? But is this thing correct? Carbon has how many bonds there? 5. So remove 1 out of this thing and put 2 here. So 1, 2, 3, 4. Do you understand it? So this is the structure of your system. The next structure is the structure of what? After system we have thyroxine. Thyroxine. Hey, look. Just look at this. Just look at this. How do you spell tire? Thyroxine. This all these things is telling us that we have 3 things here. We have 3 things. Okay, we have 3 things. Okay, please don't sleep on it. Do not sleep on it. We have 3 things. The first one is always alanine. Okay, we have OH. Abi, we have OH and let's spell tire first. TYRA. That means we have something tire. And when you look at the structure of your organic structure, which one do you think looks like a tire? Is your benzene. Your benzene actually looks like a tire. Abi, now if I want to draw the structure of a benzene now, right, it looks like a tire. If you can look it very well, it looks like a tire, or should I call it a board? But it looks like a tire to me. Okay, so we have thyroxine. So we have alanine, OH, and this benzene group. Do you understand it? So now let's draw the structure. Alanine, NH2, carbon, COOH, hydrogen, our benzene. Draw it here. No, complete the structure of your alanine. Okay, but instead of writing CH, just write CH. Just write ordinary CH, just for the blunt. Okay, let me just write it as CH3 first. Okay, draw your benzene, this thing to it. Do you understand it? Then add an OH. That is it. You can see it's not add, but this carbon is having how many bonds around it? 1, 2, and 3 for hydrogen, making 5. So we need to remove 2 bonds. Okay, so that will be 1, 2. So we have 1, 2, and 2 from hydrogen, making 4 bonds for thyroxine. Okay, now that is that about that. So now the next thing is I have asparagine and glutamine. You notice that what both of them sounds alike. Asparagine, glutamine. Now these 2 guys, AS and GLE. Asparagine, glutamine. One thing I want to tell you about these guys is that what these 2 guys, they are still part of this. They are polar, but they do not have a charge. Okay, they are polar, but being polar, they are not. You notice that? Okay, don't let us go there. I don't just want to go out of this. But this asparagine and glutamine, they are actually under a particular group known as amide. They are called what? Amide. Amide. What did I call them? Amide. I just want to give you a cheat to remember their structure. They are known as what? They are amide. And as an amide, amides have their own what? Functional group. And the functional group of amide is what? CONH2. The functional group of acid is what? COOH. But for an amide, the functional group is what? CONH2. Do you understand it? CONH2. But for an acid, COOH. So I said asparagine and glutamine are what? Amide. It means that what? You are going to draw the normal structure of your what? Of your alanine, because we said all of them will have the alanine structure. Okay, and you will put the group there. Okay, so for asparagine, it means that what? After the structure of your alanine, for asparagine, you are going to add what? Amide functional group. After the structure of your what? Your glutamine, you are going to add what? Your amide functional group. So for the asparagine, so we are going to draw NH2COOH. OOH HCH3. Now the amide group, which is what? CONH2. Do you understand it? But remove one hydrogen from this methyl just for the bonds around the carbon to be complete. So that would be what? CH2. Do you understand it? This is what? Asparagine. This is what? Asparagine. And the shortcut for it is just what? AS. The three code rather. AS. Okay. And you know why we say it is ASN? Because just pick AS from them, and because they are amide, they have amino, you can just write the N, which is for what? Amino. ASN. Okay, do you understand their structure? Now I will not clean this structure, I will draw glutamic acid. Now, how do I draw the structure of glutamine? Glutamine, don't forget glutamine is also an amide. So how do I draw the structure? Will I just draw this alanine? Do you think I will just draw alanine and then add the amide functional group? No. It means that if I should do that, both asparagine and glutamine will have the same structure. Okay, it means that just for them to be different, let me just add one more carbon to what? Glutamine. How do you know you want to add one more carbon to? This one is asparagine. This is the first thing. You don't have to add anything to it. Glutamine is the second one. It starts with what? G. So you have to add something to it just to tell you that what? G is what? Greater than A. Okay, do you understand it now? Let me use this space. Let me use here for the structure. For the structure, we have NH2COOH, hydrogen, CH3, just add another CH3, and add the functional group CONH2. That's all. So this is what? Glutamine. Do you understand? This is what? Glutamine. I hope you understand it. So, but let's balance the bond. So this one should just be what? CH2. This one should also be what? CH2. So this is your what? Your glutamine. Okay, this is your glutamine. Don't forget, glutamine and asparagine are they are what? Amide. And they are also polar. Okay, don't forget, but they are polar without a charge. Now the next one is, let's move to the structure of the what? The acidic and the basic one. Don't forget, they are also polar. Okay, just the acidic and the basic one. For the acidic, guys aren't acidic. Guys aren't acidic. Guys aren't acidic. We have glutamic acid and aspartic acid. So how do I draw the structure? For the glutamic acid, let me start with the aspartic acid. For the aspartic acid, that is to say that what this is an acid. It means that what it has another functional group of COOH. We know that what the structure of a normal amino acid has COOH group. So but for aspartic acid, it has another functional group of COOH. The same thing for the what? Glutamic acid. It has another group of what? COOH. For the aspartic acid, it's just like your asparagine, but instead of the what? Carbonyl group, instead of the amide group rather, amide functional group, you just put this what? Acidic functional group. The same thing for what? Glutamic. You just change that of what? Of the asparagine. So let me draw that of the aspartic. So NH2, let me use the black marker. NH2 COOH hydrogen. That is our alanine. Okay, draw the acidic COOH. Do you understand it? So for this to be balanced, put CH2. So this is what? Aspartic acid. But don't forget that what? Acid, because some textbook will not put the H. They will just remove the H and put this minus sign. That is to tell you that what? On the norms, acid will give out what? Proton. Don't forget. So you can put it there. Okay, for the glutamic acid, it is just this structure but with one more carbon. Don't forget, the same structure with one more carbon. Please, is this side showing? Can I draw at the bottom? Please check if I can draw at the bottom. Sorry, it's showing, right? Thank you. NH2 C COOH hydrogen. This is glutamic, okay? CH2 CH2 COOH. So this is your what? Your glutamic acid. You can remove the hydrogen and put a charge there. That is, because it is an acid, it can lose the what? Hydrogen. That is a proton. Do you understand it? So that is that about the what? Acidic. So the last one. Okay, let me just give you a clear. How do you represent this thing? The letter for the glutamic is glue. Okay, GLU for the 3-letter code and E for the 1-letter code. If you join them together, that will give you a what? A glue. Okay, so GLU is for the what? 3-letter code and E is for the what? The 1-letter code. Joining them together, just know you have a what? A glue. Okay, for the what? Other one, where is the okay? For the aspartic, it is what? ASP. Okay, why did this thing is what? D. They are always what? Following each other. After D is what? E. Do you understand? So now the last one is just the what? The basic amino acids, and we are done. So the structure of the basic amino acid. Please, is this thing clear again? It's still clear, right? Yeah, thank you. So now let's move to the last part, which is the what? The basic ones. So his lies are basic. Very simple guy. His lies are basic. The guy doesn't lie too much actually. So his lies are basic. Is leading lysine and arginine. So I will start with the structure of lysine. Okay, let me start with the structure of lysine. So let me start with the structure of lysine. Now, this 1, 2, 3, these guys are what? Are base. It means that what? After drawing the normal structure of the amino acid, they are base, and base is spelled as BASE. That is 4. It means that what? They will have 4 more carbons. After all of them, we have what? 4 more carbons. After the what? The backbone of the amino acid. After the NCC, you're gonna add what? 4 more carbons to them. Okay, so lysine means that what? Its own structure is what? Linear. Just a trick, it is linear. Others are not like that. This one is kind from the word lysine. Just know that what? It has a linear structure. Okay, just a straight structure. So how do we draw it? Our backbone. Sorry, let me use a black marker because I think the black marker is still clearer than the blue marker. So NH2 COOH, right? So this is our backbone. So this guy is gonna have how many more carbons? 4 more carbons. So 1, 2, 3, 4. Now that's not all. I can't just leave this thing like this. It does not make sense. How am I going to know that what? This is the base? Don't forget that what? We have a very common base, which is what? Ammonia. Ammonia will always what? Take in a proton to form what? Ammonium ion. It means that what? After the 4 carbons, just put what? Your what? Ammonia, NH3. But instead of putting NH3 here, you will notice that what? This guy is gonna have how many bonds? 4 bonds instead of what? 3. You can actually leave it as what? 4 bonds and put a positive sign on it. Okay, but it is not necessary. Just for the bond to be complete, you can just remove the 3 and write just NH2, so that nitrogen will have 3 bonds around it. So 1 and 2 from hydrogen, and you can write the 3 and just put a what? A positive sign. Okay, but not necessary. If you feel like doing that, you just understand your structure. Okay, so this is for the what? Lysine. Very simple guy. Now the next one is arginine. You see, the structure of arginine is kind of tricky, and Now the next structure is the structure of what? Arginine. But the structure of arginine is kind of tricky. But for you to understand the structure of arginine, for you to understand how to draw it, you need to understand something first. And this thing you need to understand is very what? Simple. You need just a basic thing. And the basic thing you need to understand is what? The structure of urea. How do we draw the structure of urea? What is urea? Do you understand it? Urea is just what? A compound that has what? 2 nitrogen and 1 what? Carbon. And what type of carbon? A carbonyl carbon. It is what? A compound that has what? 2 nitrogen and 1 carbonyl carbon between the 2 what? Nitrogen. So we say the structure of urea is, for us to understand the structure of arginine, one of the first things we need to understand is the structure of what? Urea. So how do we draw urea? Urea is what? Nitrogen with what? A carbon in between them. And what type of carbon? A carbonyl carbon, that is a carbon that is carrying what? Oxygen. Now it means that what? This sugar will be what? Attached to this what? Carbon with a particular bond. It will also, this nitrogen will be what? Attached to this carbon with a particular bond. This nitrogen will also attach to this what? Carbon with what? A particular bond. Don't forget that what? Your carbon should have how many bonds around it? 4 bonds. So how many bonds are these? We have 1, 2, 3, 4. So the bonds around the carbon are what? Complete. So now the next thing is what? What about the nitrogen? Nitrogen is supposed to have how many bonds around it? 3 bonds. But now this is 1 bond. But it's supposed to have how many bonds? 3 bonds. It means that what? We need to add 2 more hydrogen to it. That means we need to add 2 more hydrogen to it. So adding 2 hydrogen to it, so this nitrogen was bond is now what's completed now, because we have 1, 2, 3. What about this nitrogen also? It has 1 bond to the carbon, so it needs what? 2 more hydrogen. So this is the structure of your urea. Urea has what? 2 nitrogen with what? 1 carbonyl carbon between the what? 2 nitrogens, and you will complete the bond around your what? Around your nitrogen. That is the structure of urea. I hope you understand that. Now with this, you will be able to understand the structure of what? Of your arginine. With this, you will know how to draw the structure of your what? Your arginine without stress. Now the next thing is that for us to draw the structure now, you need to understand small thing again. If you actually what? A football lover, you don't have any problem. You just need to what? Remember this name, Kun Aguero, a very popular player, Kun Aguero. So this is what we are going to use to draw the structure of our urea. Don't forget that what? When you want to draw the structure of any amino acid, apart from what? Glycine, all amino acid will surely have the structure of what? Alanine first. That is one of their first structure. So each amino acid should have what? The structure of what? Alanine first. So how do we draw the structure of? Now I said don't forget this player name, Kun Aguero. So this Aguero stands for what? Arginine. Okay, just a trick. This Aguero stands for what? Arginine. This Kun is telling us what we are going to what? Add to it. Are you with me? So this Kun is telling us what we are going to add to it. So now if I want to draw the structure of what? Arginine, the first thing is what? Draw the structure of your what? Alanine, then add some other stuffs to it. Do you understand? So how do we draw the structure? When you draw your alanine, now this Kun is telling you something. Okay, now let me tell you something about this Kun. Let me just tell you something about this Kun before we go into the structure of arginine. There's something we call derivatives. Okay, we call it derivatives, that is derivatives of a particular amino acid. Just like we have tyrosine, a lot of things can be gotten from your tyrosine. You can get your dopa, you can get your dopamine, you can get your epinephrine, non-epinephrine. When I say epinephrine, it means what? Epi on top, nephrine, that is on top of your nephron, which is what? It means on top of your kidney. No epinephrine, no means that what? No, that is, it is not on top of the kidney. So that is that about derivatives. Now what are the derivatives of arginine? That is, what are the stuffs you can get from your arginine? You can get your stuff from this Kun. That is, this case is telling you that what? You can get creatinine, This U is telling you that what you can get urea, and this N is telling you that what you can get nitric oxide. So these three guys, cone is what? The derivatives of what? Arginine. K is for what? The creatinine, urea, and what? Nitric. Let's go into the structure of arginine. So I'm going to use here, I'm going to manage here. Okay, don't forget how to draw the structure of your urea. Urea has 2 nitrogen with what? 1 carbon in between them. And what type of carbon is that? It is what? A carbonyl carbon. So if I want to draw the structure of urea, one of the first things I need to draw first is the what? The alanine, right? So how do we draw the alanine? NH2 carbon hydrogen carboxylate, that is COOH, then CH3. This is the structure of our what? Our alanine. But because I will be adding something to this, so I will not write the 3, I will just write 2. So what am I going to add? This cone is telling me that what I'm going to add 3 things. I'm not adding urea, creatinine. This creatinine, urea, and nitric oxide is just telling you the what? These 3 guys are telling you the derivatives, that is the stuffs you can get from your arginine. And also this cone is also telling you that what? After you've drawn the structure, you've drew the structure of your what is it called? Alanine. The next thing, it means that what you are going to add 3 things to it, which is KUN. Okay, now you are going to add 3 things, okay, but one of the 3 things must end with what? N. K means 3, right? K, U, and N. So this K and U should be the same thing. Why they should end with N? That is, K I will represent it with what? CH2. U I will represent it with what? CH2. I will then end it with what? N. Do you understand it? So let me use a black marker just for you to see what I added. For the cone part, we will write K is what? CH2. U is what? CH2. Do you understand it? Why the N will be what? N. Cone is telling us that what? Add 3 things to the what? To the structure of your what? Alanine. Part of the 3 things should be what? 2 CH2 and 1 nitrogen. That is, everything should end with this what? Nitrogen. 3 things and they should end with what? Nitrogen. Do you understand it? So after the alanine structure, add your cone. CH2, then N. Now, after drawing this, the next thing you need to add is just what? The structure of your what? Urea. And that is the structure of your what? Arginine. Alanine, add your cone, then put the structure of what? Of urea there. And that is the structure of your what? Arginine. Now, if this urea is coming here now, okay, it will lost that oxygen. Are you with me? It will lost the what? Oxygen. So I will now have NH2N carbon NH2, right? So it is supposed to have double bond and oxygen, but it has lost this what? Oxygen. Now, because this guy has lost this oxygen, this double bond will then be found between what? Either here or here. Do you understand? You know it has the double bond before. Do you understand? It has the double bond here before. So the double bond will be found either between this nitrogen and carbon or between this nitrogen and carbon. You can actually what? Put it in any of the 2. You are still going to have the same thing. So this guy has lost the what? The oxygen. So the double bond on the carbon will then what? Come probably somewhere here or somewhere here. Okay. But now check the bonds around this nitrogen. This nitrogen is supposed to have how many bonds around it? 3 bonds, but it is having how many bonds now? 4 bonds. 1, 2, 3, 4. So that is to tell you that what you need to remove 1 hydrogen from here. Can you see the way I'm doing it? Just write it the normal way, then check your bonds. Do you understand? So it will then, this carbon will then come. You know, like we only remove 1 bond. It was double bond before, but we removed 1 bond, right? So the remaining bond that remain will be what? Placed here. So I will then place it here. Let me use the blue marker for that. So this is the remaining bond that remain. I will then place it here. This will be this. This is the bond. This is the carbon. Okay. I have double bond here. HN, and I have NH2. So this is the structure of our what? Arginine. So let me come again. The first thing is the what? The structure of your what? Alanine. After the structure of your alanine, don't forget your cone. Cone is telling you that what? Add 3 things, okay? CH2, CH2 then N. That is CH2, CH2 then N. After that, attach the structure of your what? Urea to it. But before you attach the structure of your urea to it, the oxygen on your urea will what? Go. Now when the oxygen on the urea go, you know the oxygen on your urea is what? Bounded to the what? Carbon, using what? A double bond. So when the oxygen go, one of the double bond is going to fall between one of these, either here or it falls here, okay? So you can put it in here. So when it falls, just check the number of your hydrogen and the bonds between the nitrogen, whether it is complete or not, okay? So after that, just bring this guy here, the remaining bond, one bond that remain, just attach it to the what? To the last letter of your cone, which is your nitrogen. So you will get the structure of your what? Of your arginine. Now what I have to tell you here is that what? You notice that what? This is the structure of our what? Arginine. But if I should remove this lower part, this the last part I added was this our urea part. So if I should remove the urea part, that is to tell you that what? This guy also that ends with cone, the one that ends with cone here, that is from here, this one that I'm circling here. So this guy that ends with cone, that is without adding the urea part, that is to tell you that what? This guy should also have its own name. So what is this guy name? This guy is known as what? Ornithine. Do you understand? It is known as what? Ornithine. Okay. Now adding the urea part to this will give us what? The what? Arginine structure. And I hope you understand that. Yeah, thank you. Pause that in a bit. Now the next thing is the structure of what? Histidine. So how do we do the structure of histidine? So how do we do the structure? It is very simple. From this that is to tell you that what we can actually what get from this H is telling us that what we have it out here, okay? But now mind you, don't forget that what in the structure of our tryptophan, we said in tryptophan we have 3 things. We have the alanine, we have the fan, which is the phenyl, the phenyl, and we also have what we said we can change it to what? Tryptophan. That is to tell you that what we have a hot in our farm, and the hot in your farm, don't forget that what the hot in your farm will only have what? Just a single window. That is a single window. Fine. So now, but for house now, most house should have what? At least 2 windows. Do you understand? So your house here will not have just a single window. If you have what? 2 windows. That is to tell you that what? If you have what? 2 nitrogen, which will serve as the what? Window, 1 nitrogen here window, 1 nitrogen here window. It means that what? How do I draw the structure of histidine? You draw your structure of histidine by drawing your what? Alanine first, and after the alanine, you put a house there. You put a house, and that house should have what? 2 windows, and the windows are what? 2 nitrogens. Do you understand? It is different from the one, you also have the house in what? In tryptophan, but we call it tryptophan. So the house in your farm is just like a hut with just a single what? Window. That's why we only have what? 1 nitrogen there. This is just the trick. I'm not telling you that what that is why they build the structure like that, or it is just a trick. Don't forget. So now let's go to the structure of histidine. So the first thing is what? The structure of alanine. That is alanine plus house. In your house, you have 2 windows, which is 2n. So how do we draw the structure? We say NH2 CH so alanine CH3. But because I have to add something down, which is the house, so I need to remove this CH3 and replace it with what? CH2, so that the number of bonds around my carbon will be completed. So I have 1, 2, 3, 4 from hydrogen. I hope you understand that. So now let us draw the house. So how do we draw this house? So we say our house is this. So how do we draw the house? So this is your house, okay? So I hope you can see this. So this is your house. Okay, now so don't forget that what here there is a carbon, right? There is a carbon here, but this is where you have your what, your windows. Here you have a window which is what, nitrogen. You also have a window here which is what, nitrogen. So your house has how many windows? 2 windows. Do you understand? This is to tell you that what, you have a door here. Okay, this bond here, you have a door. Okay, now let's check. So for one of these nitrogen, you just put a what, a double bond. You can put it either here or here. Do you understand? But let me just put it here. So that means that what, how many bonds do we have around this nitrogen? We have 3 bonds, which is 1, 2, 3. So it means that what, I don't have to add any hydrogen to this what, nitrogen anymore. What about this nitrogen? How many bonds do we have around it? We have 1, 2. It means that what, this guy needs what, 1 hydrogen. I hope you understand it. And so this is the structure of your what, of our histidine. The first thing is what, draw the structure of your what, alanine. Draw the structure of your alanine, then put your house. So your house should have what, 2 windows. Now that is that about the structure of the 20 amino acids. So now, so just now let's look at this. Don't forget, for the essentials we have what, private, team, whole. So we have 10 essential amino acids in what, in children, but in adults this arginine is not there anymore. The P is for what, phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine and lysine. Please don't thyroxine is not part of the essential amino acid. Why? Because your thyroxine can actually be gotten from your what, phenylalanine. Because thyroxine can be produced from what, phenylalanine. Phenylalanine is an essential amino acid. So after taking your phenylalanine, you can produce your thyroxine from there. Don't forget that what, your phenylalanine has what, alanine and what, phenyl. But your thyroxine has 3 structure, alanine, thia, which is also what, that phenyl, and OH. So the only difference between the phenylalanine and your thyroxine is just OH. It means that what, that thyroxine can actually be produced from your what, phenylalanine. And for the this thing, the polar, for the polar we have set as CT GLE. These are what, polar, but they do not have any charge. We also have those that are polar but they have charge. Those ones are divided into 2, which are what, basic and acidic. So we have guys aren't, that is, guys are not acidic. So for those ones that are what, basic, we have what is lies are basic. So for the non-polar we have glad, all vampires left the island. So and we ask the question, why do you think why do you think they left? Because they left because they met a professional public health, which is shortened as what, H, public health trainers. So thank you for watching this video.